Hello everyone, and thank you for attending today's webinar. On behalf of BIS Research, I'd like to welcome you all for our webinar on syndromic testing, a new take on diagnosing infectious diseases. I am Pratiksha, lead analyst in healthcare vertical at BIS Research, and I'll be your moderator for today's webinar. So, BIS Research is a leading global market intelligence, research, and advisory company that focuses on emerging trends in technology, which are likely to disrupt the dynamics of the market. So, for today's webinar, we are having three speakers with us, which all are esteemed achievements and valuable expertise in syndromic testing. So, firstly, we are having Mr. Bruno with us, who is a country lead at BD for the BD business in Portugal. During his experience in the medical business, he has had all the opportunities to design, evaluate, and implement diverse market access, commercial, sales, and marketing strategies for a wide range of products within the IVD market range. So, this includes business in syndromic testing products also. Further, with us, we are having Mr. Emmer, who is a business development specialist in LabCorp. He's a biotechnology specialist with experience in NIPT, NGS, and molecular diagnostics. Lastly, Ms. Riya Gupta, who is working as a research analyst in BIS Research. So she has successfully handled so many projects and market assessment-based analysis to the healthcare field. I thank you all the speakers for your gracious presence today. So further, moving to the next slide, I'll discuss the agenda of today's webinar. So Bruno will be discussing all the new standards of care in the treatment of infectious diseases and syndromic testing. Then Emmet will be continuing with the use of NGS in metagenomics for acute care syndromic testing. Then Ria will be concluding the speakers webinar session for syndromic testing market growth. She will be discussing the major factors which are driving the growth of the market, some of the major players in the market, opportunities, and then after that, I'll conclude the session with a question and answer. So for the next 40 minutes, we all are keen to learn from all your experience with syndromic testing. And towards the end of the discussion, we'll open the panel for the audience to ask any question. I'd like to request everyone in the audience to kindly type the question in the chat, and towards the end of the webinar we'll take up all the questions. But in any case, we will be not be answer to any question, so that will be reverted on blog post or through email. So now over to you, Bruno. We'll start with the session.
Hi, so good morning, good afternoon, good evening to you all. It's a pleasure to be here with you today. The title of my presentation is already somewhat provocative. So when we define new standards of care, they are always challenging, and there's always two views to the same path. But that's something that I would like to address today with you, and thanking also BIS Research for the kind invitation. Next, please. So as a disclosure and disclaimer, so I'm the BD country lead for Portugal, as was mentioned. BD is a medical device company with the solutions in the molecular diagnostic market. But once again, the opinions that I will share with you today are my personal opinions and not involving BD in this overview. Next, please. The agenda for my presentation and addressing this new standard of care, I'll try to cover these four main topics. What is the need? What could be the solution? What are the proposals that are available in the market? And finally, the value that these new solutions, these new proposals can bring to the market, to the diagnostic market, to better benefit the patients, the healthcare workers, and society as a whole. Next, please. So what is the need? Next. Well, up till some years ago, lab diagnosis historically was performed using a mixed bag approach, like going to the store and picking up a a lot of different stores, a lot of different purchases, and some of those approaches were mainly used historically in old diagnostic tools. Let's talk about microscopy as an example. And when I talk about microscopy, it can be the traditional one, a direct one, or using even stains to help to visualize. Antigen detection, either the traditional agglutination methods or even the immunochromatographical ones. Culture, the Petri dish-based culture with the plated media, liquid enrichment approaches, or even cell-based culture to detect the presence of viruses in the sample. And finally, the PCR test. Well, PCR tests and the PCR technology represented a huge step forward in the diagnostic of the infectious diseases because it helped increase the speed of the diagnostics and helped increase the sensitivity and specificity of the diagnostic. As you can imagine, using microscopy-based methods, using culture-based methods, or even antigen detection methods, they have somewhat limiting values in sensitivity and specificity. PCR helped already to address these limitations, I put it that way. But to some extent, it also didn't give us a full picture about the sample, about the pathogens, because what is the difficulty is that it's very much individual, or in certain cases, duplex molecular methods at start, where we search for a specific pathogen and not for a specific condition. And that was one of the difficulties that were used molecular methods, is that we always, at the beginning, searched for a specific pathogen and not focusing on the condition itself. And in the next slide, let's have an overview about what is the methodology using a specific condition. Let's focus on gastroenteritis. In gastroenteritis, also to escape a little bit about the traditional infectious, the traditional respiratory diseases, because I think that after COVID, we are already a little bit, I would say, fed up with it, but let's talk about a different thing. Regarding gastroenteritis, rapid testing is also vital because, next please, with gastroenteritis, we need to establish also with a rapid testing the appropriate treatment for critical patients. And the appropriate treatment is very much dependent on the etiological agent that is causing this gastroenteritis. And next, please. And also to prevent the spread of the gastroenteritis through isolation measures. When we talk about an infection like a viral-based gastrointestinal infection that can easily and quite rapidly extend to other patients, or in the case other people, and we need rapidly, with a quick and a rapid identification of the etiological agent, to address the need of establishing at least these two measures. Next one. Well, and also in gastroenteritis, we also had an overview about the traditional techniques. Let's talk about the conventional diagnostic techniques for gastroenteritis. Next, please. Well, we use culture manually for, can help us in the identifying if it's a bacterial agent that is present there, for instance. Next one. A microscopy, that also with a direct microscopy, it can help us to detect parasitical etiological agents for this gastrointestinal infection, for instance. The next one. And also the antigen detection that can also be used for parasitical, for microbial, and also for viral etiological agents. So, but these are the traditional techniques that still have some of the limiting clinical value that I discussed previously. Next slide, please. And we can see it it had multiple steps. Next slide, please. And had up to 3 days for results. So here we have really a laborsome techniques, and yet some doubtful research, doubtful results, and also the length of the diagnostic. When we say that rapid testing also here is so important. Next slide, please. Let's focus, for instance, in the Campylobacter isolation. Next, please. We have a microscopy using a stain-based method with limiting powers of sensitivity and specificity. Next, please. Then we move to the traditional plate-based methods that can also involve enrichment with also some additional work that needs to be done. Next, please. And even these plate-based methods need to be have that specific needs in terms of environmental and temperature conditions to be taken, to be undergone. Next one. And finally, even in certain cases, we can need to resource to additional manual testing. So once again, laborsome, long, and doubtful results that with low specificity, low sensitivity, can lead up to uncomfortable clinical decisions that need to be taken or can be taken. Next slide, please. So what if we could simplify the infectious disease diagnostic and testing with rapid results, with a reduced hands-on time, with greater specificity and sensitivity and flexibility for targeted diagnostics? Next slide, please. Well, this leads to the solutions that were developed addressing these wishes. Next slide, please. So basically, what we tried, or the industry tried, was really to develop methodologies that could reduce the hands-on time. In the case of the PCR, even in certain cases, grouping the 3 traditional steps of the PCR with the extraction, with amplification, and in the detection in a straightforward approach, or even integrated into the same methodology. Also with a direct pathogen detection, even diminishing the labor that needs to be done processing samples. Better clinical performance using the characteristics of PCR as the sensitivity and specificity, and reducing, of course, the time to detection, and then addressing this reduced time to detection. Next slide, please. So the proposals, next. So in the market, we have already a lot of different providers in the market, of the GeneMark, Luminex, that now it's a part of Diasorin, bioMérieux with BioFire, QIAGEN with QIAstat, BD with the BD Max. But generically, they try to address these needs that we already discussed. So to be rapid, to be a fast detection time, fast answer to result, a sample to results time, to have a broad coverage, because if we focus on the condition and not in the etiological agent, we need to be sure, to the best of the extent and the knowledge that we have, that we cover, if not all, at least the vast majority of the etiological agents that cause that condition. Can be viral, that can be bacterial, and that can be parasitic, for instance. And this is very important, to have this broad coverage, to have an answer that is representative about the etiological agents that are involved in that condition. Next, please. Now addressing the value of this solution. And now I start with a little of a provocation, if I may. Next slide, please. So faster, bigger, must be better, right? Next slide. So let's now, and here to share with you, also to make us think and reflect about this, a specific case, a clinical case, just to share with you here today. And if in this case we have a case of a 80-year-old male that presents in November to his primary care provider with a 3-day history of fever, cough, headache, and myalgia. So the question is, having this clinical case, should a syndromic panel be considered in this case? So I think you can react with hands up or with clapping. If you react with a hands up, it would be yes, and with clapping, a no. Let's just give 5 seconds for people to answer. Okay, so it's more or less 50-50. Let's move to the next slide, please. Well, here, and this is somewhat what I think needs to be our responsibility when addressing this issue, and I'll share with you later in the conclusions that specific facts. Also, syndromic testing need to have a targeted use and a targeted indication. In this case, in an otherwise healthy individual without any severe disease, without any chronic situation, without any kind of other conditions, targeted testing would be most appropriate. So, and here in this case, let's go for the 2 traditional etiological agents, one viral, that it's the flu A/B virus, the other bacterial, the strep A, Streptococcus pyogenes A etiological agent. And of course, nowadays with the COVID-19, also that it's a little bit part of the day-to-day diagnosis of respiratory disease. But it should be, in fact, considering this case, a targeted approach. But the value of these tests remains very much high and very much important. Let's go to the next slide, please. So here I try to summarize a little bit an overview about what could be the limitations, and on the other hand, what could be the advantages. But in fact, it's not that straightforward, limitation in advantages. In fact, I do believe that we need to evaluate is a cobweb, as you have in the middle, of issues to address. In the top part, the ones that are more intrinsic to the lab or to the hospital, that very much depend on the scientific knowledge and technology, and in the other hand, the practical needs internally. So where you can think about new techniques, new diagnostic methodologies, and I think the omics are very much in the discussion nowadays. And on the other hand, the practical needs of that hospital, about that healthcare unit that need to be addressed. And in the bottom part, really the extrinsic or the external factors that can be a part of the laws and regulations of the state, of the country, where you are. And then the financial factors that can very much depend about the funding that can be originated to pay for this kind of approaches. But let's then move to the limitations. Well, regarding limitations, and this is a little bit an overview about what I found out in the bibliography. In some cases, these are still add-on tests. They do not allow customized ordering because they come with a panel, and some sometimes throughout the world, the panels are all the same, not allowing certain countries to customize according with their specific etiological agents that are present there. They are still costly. And when I talk about cost, I talk mainly about the price, because in the first overview, they can be with a high price, but on the other hand, they can have big and important value to the healthcare workers, or to the healthcare facility where they can be used, because they can be cost effective if multiple routine methods would be ordered. In the case that we saw before, the traditional methodologies are very laborsome. They take a lot of time. If the patients are admitted in the hospital, lengthy and costly hospital stays enter into this equation, so they may reduce the downstream costs. They don't always yield the identifications, because if there is an organism that by any chance is causing the pathology and it's not in the panel, probably there can be lower sensitivity for the detection of certain pathogens. When I talk about lower sensitivity, in some cases they may not be indicated, because if you have in a panel a pathogen that it's present, for instance, in the case of the respiratory panel, that it's mainly present in the low respiratory tract, and if you only search the upper respiratory tract, we will never find it, because it's not there. So it's not lower sensitivity symptoms, is a limitation of the test. Polymicrobial bacteremia can be challenging, and the significance of the detection of multiple targets in these multiple panels remains unclear, because the molecular and the genetic material can be shredded throughout a long time. So sometimes we can detect non-viable genetic material from microorganisms. Genetic material from microorganisms that are no longer viable, and that may yield false positive result. This is something that are the limitations, but let's then focus in the advantage. They can give you rapid results. They can allow for the detection and identification of viruses, because viruses, it's very much difficult to detect them using traditional culture-based methods, for instance. So they are very much important. And then if we detect if it's the etiological agent is a virus and not a bacteria, we can then rapidly de-escalate the antibiotherapy, because, of course, as you all know, they are not effective with viruses. We can then also decrease the use of invasive sample collection techniques that can harm, in certain cases, the patients, have and allow informed and timely decisions to be made regarding infection control measures and timely outbreak investigations. The faster that we act, the better usually are the outcomes. And then these syndromic panels, without any doubt, should be considered as the first line of diagnostic approach for the immunosuppressed population, and of course for the clinical ill patients. And finally, next slide, please. As a summary and as a conclusion, I would like to share with you that in my understanding, syndromic panels, if implemented thoughtfully and interpreted carefully, really have the potential to improve a lot of patient outcomes and even the antimicrobial use, because they allow to improve the clinical decision-making process, to optimize the laboratory workflow, and enhance the laboratory and antimicrobial stewardship of the labs. And finally, as clinical experience with new syndromic diagnostic platforms continues to grow, it will be important also for clinicians and even for the industry to share their experiences regarding the implementation and optimization strategies. And the final question that I have here that needs to be always taken into consideration by everyone that is going to use these kind of panels is, will I manage my patient differently based on the results of this test? What if I get a positive? Thank you so much to all of you. And next slide, you have my contacts, and I'll be more than glad to answer some of the questions that you have further on. Okay? Thank you.
Thank you. Thank you so much, Bruno. So this was indeed insightful for syndromic testing. So Amar will be not joining us on the call due to some emergency. So now Ria will be taking over the session. So now Ria, over to you, please.
Patricia, yes, thank you. Thank you. So I'll start with introducing syndromic testing, the key trends in market development. So we at BIS Research have already done a lot of reports on infectious diseases and are trying to keep up with the technological progress it is making. So our latest report was, of course, about syndromic testing, since it's the latest technology in infectious diseases right now. And the CAGR that we are expecting for syndromic testing in the coming years between 2023 to 2033 is a 9.43% growth rate. If you look at the different regions where the tests are being adopted, most of the tests come under the low share, high growth quadrant, which is the bottom quadrant 4. So this is because this is a comparatively new technology, and it is being adopted around the world pretty quickly. The highest share is from North America. This is because they have adopted it the highest so far, and then closely followed by Europe. The rest of the regions, Asia, Pacific, Latin America, MENA, and rest of the world, are in the high growth area, but they are still to adopt. So they are in the process of adopting really fast. In the coming years, though, we expect that the syndromic testing market matures, as it is currently a very nascent market, syndromic testing will become the key test for screening infectious diseases. So now let's talk about the testing landscape based on the different test types. So as you can see in the pie chart, the most common test type right now is respiratory, followed by gastrointestinal, genitourinary, and tropical diseases. Other diseases include nervous system-based diseases and blood culture-based diseases. So these panels test for a bunch of diseases. So in the respiratory panels, of course, are the most famous right now, most popular right now, because of the COVID-19 pandemic mostly. And we expect that it will increase to hold around 56.2% market share by 2033. So right now, just the respiratory syndromic testing panels alone make up of 4.74 billion of the market by 2033. So moving on to the next slide, these are the big players, the key dominating players in the syndromic testing market. Most players in the market, as you can see, are big diagnostic companies, including like bioMero, Sagene, Cepheid, and Luminex. Bruno also mentioned some of these companies. These companies have a market share of greater than 6%. Then there are companies like Abbott, Kaigen, Hologic, Roche, which hold a market share of around 4 to 6%, as they do other things than just diagnostic testing. And then there are companies like Biorad, BD, Siemens Healthineers, which are also coming up, increasing their market, this syndromic testing approach, and will be able to dominate the market in the coming years. Now let's move to the key dynamics of syndromic testing. So There are a number of factors that are growing the demand of syndromic testing in the market right now. So according to our study, the key factors include increasing incidence of infectious diseases, the early diagnosis of syndromic testing, and the reduced use of antibiotics because of syndromic testing. So let's talk about the first factor, the increasing incidence of infectious diseases. In fact, the world right now is facing a lot of problems, including the increasing population, the temperature change because of global warming, urbanization, so people living closer and closer together like they've never lived before in the cities. And there's also the accessibility to places now. So people can travel so much faster because of airplanes. Air travel has increased drastically over the last 50 years. And because of all these factors, the spread of viruses and diseases has become really, really common now. With increased temperatures, the transfer of viruses is faster as they get a better climate to habitat for their habitation is better. And all of these things are increasing to increase, leading to the increase of infectious diseases. And so in the coming years, as infectious diseases increases, we need to find a way to combat them before they become a really big thing. So this comes to the early diagnosis that syndromic testing can provide. So if the patients, like Bruno also mentioned in his presentation, so patients are diagnosed at a much earlier stage, which also reduces their healthcare cost and also just their life in general. And the third one is the reduced use of antibiotics. People are getting addicted to antibiotics more and more nowadays as, they become easier to get. This will also reduce as if the number of infections increases. And this is also explained better in the next slide. So I'll go to the next slide. So this here, the figure on the left shows the time of diagnosis. So since this first start of diagnosis and the light blue color showing the last stage of diagnosis. So as you can see, the singleplex test, the results come right at the last stage of diagnosis. So much time has passed after the onset of symptoms when you actually go to get that test and get the results. As compared to with syndromic testing, you can get tested at the very early stage, and that will just change the way you are being treated. So on the right now, we have the reduction of antibiotics use. So these are a few different studies, and the results of these studies say that there will be a 13% reduction in antibiotic therapy in the coming years, a 30% reduction in antibiotic prescriptions because of syndromic testing, and so less in the median antibiotic during adult influenza patients. So all of these things will be possible with syndromic testing. So that was my presentation. Thank you. Thank you, everyone. Shall we move on to the next slide now?
Yeah, sure. So Ria, Amar is here, so we can continue with his part also.
Yeah, I'll just, okay.
That would be great. Yeah. Hi, Amar.
I think he's on mute right now. Amar.
Nikita, can you just
Hello, everyone.
unmute him? Yeah. Hi, Amar.
Hello, everyone.
So now Amar will be continuing with his part, like his insights for diagnostic infectious diseases for syndromic testing. Amar, you can go ahead.
Okay, thank you. Hello and welcome, everyone. Thank you for attending the live or the recorded version of this webinar. I am excited about the topic and being part of the discussion of the advancement of an important topic in diagnostics. I am interested to learn how healthcare providers, laboratory professionals approach syndromic diagnostics and what measures you take when you face challenging infections. So in the following presentation, I want to talk about how next generation sequencing can enhance syndromic diagnostic capabilities and get the latest updates on the advancement in the realm of diagnostics, and ultimately enabling you to make an informed decision in future investment to ensure providing excellent care for your patient. As metagenomic NGS has the potential to improve your patient outcomes through improved clinical decision-making, optimized laboratory workflow, and enhanced antimicrobial stewardship. So for the first slide, I want to talk about, throughout my presentation, I will go through an overview over the infectious disease. What are we doing today to in diagnostics? And what is the metagenomics NGS? What are the key applications of this technology? And a summary on the end of the presentation. So first, in the overview here, we all know infectious disease are caused by pathogenic microorganisms such as bacteria, viruses, parasites, or fungi. The disease can be spread directly or indirectly from one person to another. Sources of infection range from contaminated food and water, interacting with animals, or working on hospital and clinical setting. So in a world where time is of the essence, advanced diagnostics play a pivotal role in delivering timely treatments. So next slide, please. So the global burden of antimicrobial resistance continue to rise. Antimicrobial resistance poses a major threat to human health around the world. The infections are harder to treat and increase the risk of disease spreading severe illness. So we have from the World Health Organization, the 5 priority. The next slide, please. I'm sorry, the previous one. Okay, so in these times, the judicious use of antimicrobial is of utmost importance in these times. We have to moderate the antimicrobial use in clinics because we face new things like novel unknown infections, and we have pathogens mutation that are resistant to these known treatments, and also not to mention the outbreaks and new viruses like what we had in 2020. So now the next slide, please. Okay, so the drug-resistant infections affect lives of billions worldwide, and actually it's approximately reaching $1 trillion additional healthcare cost by 2015. So this is a heavy burden we have. So next slide, please. So what are we doing today to diagnostics? Advanced diagnostics can be divided into several groups according to their methodological approach as well as their practical application. We have the conventional pathogenic techniques, proteomic, primarily using matrix-assisted, laser absorption, ionization, time-of-flight mass spectrometry, molecular assay targeting specific genes, and genomics or metagenomics. The traditional clinical paradigm for diagnosis of infectious disease in patients applied for more than a century involves a physician formulating a differential diagnosis and then ordering a series of tests, generally one bug, one test, in an attempt to identify the causative agent. With culture-based diagnostics applicable mainly to bacterial and fungal pathogens, one or more culture phases are involved to yield growth of the suspected microorganisms from clinical samples. Subsequently, growing isolates are characterized with respect to taxonomy, antimicrobial drug susceptibility, and traits such as virulence and molecular subtypes. These mainly include molecular assays performed on culture isolates also consist of polymerase chain reaction amplification and detection of specific genes from, for example, those inferring antibiotic resistance and amplification of the 16S ribosomal RNA gene, and subsequently using Sanger sequencing. And I will talk about why we target this, particularly this region in the genomic, the 16S ribosomal RNA. And finally, and here we have the single cell whole genome sequencing, powered also by NGS, is performed by sequencing in parallel in a very high number of bacterial DNA fragments, followed by bioinformatics analysis to reconstruct the fragmented DNA sequences back to contiguous genome. On the other hand, what we have today as the future is the culture-independent microbiology. Involves the application of diagnostics techniques directly on the clinical samples. The main one is the application of PCR assays targeting specific genes that relate to the presence of pathogens and important inferred phenotypes such as also antimicrobial resistant to a key agent. And more recently, a massive increase in the availability in-house and commercial multiplex PCR assays is evident, as Bruno mentioned, covering a wide range of diagnostic target in a single run. These assays are increasingly designed for syndromic diagnostics, including the most common pathogens causing infections in the gastrointestinal and respiratory tracts. When we're discussing omics-powered diagnostics tools, however, the focus is on the application of NGS technology directly on the samples, an approach also known as metagenomics. So next slide, please. So what exactly is metagenomics? Next generation sequencing? Well, it's the discipline that refers to the sequencing of all nucleic acid material present within the clinical specimen or to recover clinically relevant microbial information. It is the comprehensive analysis of microbial and host genetic material at the same time, the DNA and the RNA in samples from patients. And to illustrate that, I think of it as when I think of metagenomics, I think of it as if you had a box of puzzles, but it's not just one puzzle. You actually have all 10 smaller puzzles put together into one box. And when you want to think about metagenomics and the genomes of these 10 organisms, let's say, you are trying to solve 10 puzzles simultaneously to understand all the different pictures that are in the same box of genomes or sample. So it doesn't merely identify one or two pathogens, it detects a multitude of potential targets simultaneously, expediting the diagnostic process and streamlining the course of treatment. The previously daunting task of identifying the source of symptoms is now rapid and comprehensive endeavor. For patients where conditions where co-infection are clinically relevant, metagenomics can provide a powerful diagnostic tool to help inform and guide care. So next slide, please. Thank you. So what the historical overview of metagenomics, how we get here? Well, for almost 300 years, the study of microorganisms can, based on morphology, features, growth rate, and selection of some biochemical profiles. And in the 1970s, scientist called Carl Woese proposed the use of ribosomal RNA genes as molecular markers for life classification. So the idea, in conjunction with Sanger automated sequencing method, revolutionized the study and classification of microorganisms. Later, the advances in molecular techniques were applied to microbial diversity description and granted access to a new uncultured world of microbial communities. And some of these techniques, PCR, ribosomal RNA genes, cloning and sequencing, fluorescent in situ hybridization, denaturation, gradient gel electrophoresis, restriction fragment length polymorphism, and terminal restriction fragment length polymorphism also was used. However, in spite of all these improvements, there were many other observations in microbiology that remained unanswered, like those related to microorganisms, metabolic and ecological functions, characterization of certain functions in a particular environment was possible only after gene cloning from total DNA of a certain habitat, and when its heterologous expressed products was associated with a given metabolic function. So this implied the advancement of gene expression techniques using other microorganisms as systems to test gene function and roles in the microbial community. So we get to discover new genes using sequencing to discover new genes, functions, and metabolic products with technological application products such as we use today as broad spectrum antibiotic. Antibiotics has set the foundation to a new era named metagenomics analysis, which was later defined as the theoretical collection of all genomes from members in the microbial community from a specific, let's say, environment or a clinical sample. So this emerging approach has changed how physicians diagnose treat infectious disease, with applications spanning wide range of areas, including antimicrobial resistance, the study of the microbiome, the human host gene expression, what's called transcriptomics, and of course, oncology. Next slide, please. Okay, I will try to be short here for our time. So Sanger sequencing, although used to completion of the Human Genome Project, it's a laborious, time-consuming, and expensive techniques. Actually, the need for rapid, accurate, and what's called high throughput generation, generating multiple results during single machine run, give rise to a new sequencing method, the next generation sequencing, such as the Roche Pyro sequencing system in 2005, and later on the various platforms produced by Illumina in MySeq, HiSeq, and NextSeq. So here we have the metagenomics next generation sequencing types. We have in targeted metagenomics, the sample is subjected to an amplification step, usually of the 16S, again, ribosomal RNA gene. Why? Because as it has proven to be a useful molecular target since it's present in all bacteria, either as a single copy or in multiple copies. And it's highly conserved over time. So it's you can say it's sterile site in the genome of this pathogens or in the pathogens genome. So this method is mostly used to describe the microbial population in a body site or the term microbiota. So another approach is applying metagenomics, and in a wide range is a shotgun or whole genome. I see we are still in the previous slide, please. I will continue, no problem. For applying the metagenomics such shotgun whole genome metagenomics, in which there is no pre-sequencing specific amplification phase, and the entire genomic content of a sample is being sequenced without introducing biases. So using shotgun metagenomics enables the identification of all microorganisms present in a sample, including viruses, fungi, and parasites, as well as the characterization of other important elements such as antibiotic resistance, determinants, and virulence vectors. So for the key application of this technology, we have general application here, clinical applications, epidemiology application, animal health monitoring, food safety, surveillance, drug discovery, and environmental studies. In this slide, the key application of the clinical application, this is a non-exhaustive list I put here, including sequencing and discovery of novel genes, enzymes, new species discovery, pathogens diagnostics. In the next slide, please. Yes, the pathogen diagnostics, and also tremendous pharmacogenomics applications and antibiotic resistance. So the skipping the cultivation step, we can say clinical metagenomic could constitute a rapid and generic approach providing all medically actionable information. The presence or the absence of microorganism, their identification to the species level, or the detection of antimicrobial resistance. Okay, next slide, please. Okay. So to implement the NGS, we have two sections here. The integration of these platforms are in the microbiology workflow. Commonly includes the wet lab laboratory, which includes the DNA extraction, library preparation, where extracted DNA is randomly fragmented into same size pieces, and then ligated to primers and adapters. And then template preparation, including amplification, sequencing, which in the case of sequencing by synthesis methods involve the incorporation of fluorescent labeled deoxyribonucleotide triphosphates during each cycle of DNA synthesis, followed by the identification of fluorophore excitation. The second part, and the complex one, is the bioinformatics, which includes the analysis of the sequence data and translating them into easy to understand reports, and to actually classify and identify the microorganism we are studying. So as we reach the end of the topic, I want to summarize. The metagenomics has a broad applications in various industries. from studying environment, identifying infections, and to identifying antimicrobial resistance. And so we anticipate that the use of metagenomics NGS in medical microbiology laboratories will further increase over the next few years for the characterization and surveillance of pathogen, the investigation of outbreaks, as we see useful application of that, and infection prevention and the detection of novel resistant genes. Also, the whole genome shotgun sequencing is a powerful tool to study microbiome, and soon will have a significant impact on health and disease as reflected by the growing market. So for everyone today, I would like to thank you for attending and also express my gratitude to Bruno for his insightful presentation and BD remarkable capabilities, along with everyone here from BIS for the hard work they put into organizing this webinar. So thank you.
Thank you so much, Emil. So this was indeed very insightful for all of us. So again, I am thanking all the speakers for your valuable insights on the syndromic testing. So now moving forward, Riya, next previous slide, please. That sample one. Previous, please. Previous. Okay, so in case you wish to see our sample of any market intelligence report, you can easily ping us on the Teams, and we'll get back to you very soon. So next slide, please, Riya. Yeah. Now coming to the Insight Monk. So the whole concept of designing for coming up this Insight Monk was to share the knowledge with industry experts that we have. So at BIS, we are successfully conduct these niche projects which are like heavily rely on the AI-based platform mainly. So further, the web platform is beautifully and designed depicting the three major sections that could be of interest. First is where like industry experts can try to integrate market experts from different industries, from different regions, that they can come together for the web platform where they can network with each other. And other section that is very insightful regarding the industry reports, which would be collated from various reliable sources. And lastly, there would be like question answer section where you can engage in the existing discussion or initiate your own discussion. So further concluding this session for today, I want to open the panel for any question from the listeners. I request the listeners to kindly type the questions, and I'll be happy to pass the questions to the esteemed speaker. Or if you want to ask the particular question to any particular speaker, then please let us know. We'll forward them. Yeah, so.
We have a question in the chat if you want. I can address it. I don't know if you want to compile.
Yeah, sure, Bruno. Please go ahead.
Yeah. Back to it. Well, Amy, thank you for the question. The question is, what are the challenges with which are currently being faced for the adoption of syndromic testing? Well, I try to address, and please join in if you want to add also. So I try to address some of them in the limitations part, but in fact, I think it's really a cobweb of challenges. I think that here it's very much connected with the scientific, the practical needs of each institution, the laws and regulations of the places that they are undertaken, and then of course the financial factors at the end. I think, as always, to the adoption of new technologies is always a balance between the resistance to change, that is intrinsic to all of us. And then at the same time, some difficulties in adopting it, mainly the cost. I think this transition for costs to value is something that needs to be undertaken by each healthcare professional, by each healthcare institution, and even for each of the financiation agents, being the state, being insurance companies, whatsoever. Because at the end, what we see is, although at the first hand, these kind of tests can be costly, if they are targeted correctly, if they are addressed in the correct patient at the correct time, they really are cost effective. And I try to show you that in certain cases already, they should already be the standard of care.
Yeah, thanks. Yeah, thanks, Bruno. So I think so we are having another question. So what is the reimbursement maturity for syndromic testing? Amar or Bruno, anyone can take up this.
I don't know here, to be honest, I'm a little bit with maturity. What do you want to, what can be the case reimbursement maturity is when are they paid? What is the logic for reimbursement? I don't know what you mean with that, Sean, sorry.
Okay, so there is another
if the
question.
payers cover
pay
this
us
testing.
Yeah.
Okay.
Yeah.
Well, I can speak about the case
Sure.
that I'm more comfortable with, that is the case of Portugal. Here we have mixed models, so we have outpatients that usually go to private labs, private lab chains, and usually they are more based on single test, so they are paid per meter per radiological agent, not as much as syndromic diagnostics. The vast majority of these tests, at least here in Portugal, are being undertaken in public hospitals. In public hospitals, government takes care about full payment of these tests, and they are mainly done for specific conditions and also for specific patients. So usually these tests are done mainly in hospital settings. So the government, that is the main funding agent here, takes care about them. If, of course, if they want to take the test, and well, Amber here can also add on it. Big lab corporations also have this test available. I imagine that some insurance companies can take care of that payment. Here in Portugal, there is not that tradition, but even out-of-pocket payments can be done.
Yeah, thanks, Bruno. And yeah, again, we are having another question. So how do you see the role of metagenomics for the upcoming 5 years in the syndromic testing? I think that this is for Amber. So Amber, can you please answer this?
Yes, sure. Thank you for a great question, actually. Well, it depends, because there is a lot to consider when a clinical laboratory decides to incorporate this technology in their labs, because there is a huge, in some degree, huge investment to be done to the, as I said, in the dry, in the wet lab routine and the dry, the bioinformatics section of the lab. And there is a huge investment will be done there to incorporate this technology. So what is the challenging things in this case is we want to sequence the genome of this infection, and then we have to compare it to what we have already in in genomics databases. So we are at least at my this is my view. We need more time to enhance the databases that contains the pathogenic genomic information to be comfortable to say, you know what, we can now rely on this databases to actually identify pathogens and use it as first tier technology. So we will have, I think we need more time to enhance this databases, and we have to invest in education especially for the bioinformatics field, because there is a serious lack of personnel there. It's an intersection between biology and let's say informatics or programming or coding. So it's difficult subject, and we don't have enough personnel to actually do this job.
Okay, that was quite insightful, I think so. So, are we having other questions? Or should we wrap up the session? If you guys are having other question, you can mail us, or yeah, that will be easy for us. Then we'll answer on that. So thank you all the speakers. Thank you on the behalf of BIS Research. It was a great webinar. Thank you all again. Have a great day ahead.
Thank you, everyone. Thank you, Bruno. Thank you, Pratiksha. Thank you, Raya. Thank you, everyone, for attending this webinar. Thank you for the.
Thank you all. See you next time.
Thank you, everyone. Thanks for joining.
Thank you. Bye-bye.
Goodbye.